people who are so good at their work have taken the time out to be with us, particularly on a Friday evening. Appreciate it. I understand that a lot of them have attended the previous talk as well and have a few questions for us. We are looking forward to it. I can't help resist but say that I'm slightly nervous. All of you are from the clinical side, and we look at the data. Hoping that we are able to answer questions and also improve ourselves and learn a lot from you all as well.
True, I totally agree with you. And that's the reason this particular session was organized. In fact, there can be a lot many questions. I want to make it clear to the participants as well, to the professionals who have joined in, that yes, your questions, it's a win-win situation for everyone. Whether your questions are answered or are left unanswered, the most important thing is we are putting it out so that we can start working on it, because those are the hot things that we can work on. Your inputs make a lot of difference to all of us. So whatever it is, I would again request each one of you to please put in your questions across. You can, if you want me to speak your question out, please put it on the chat box, and otherwise you can always unmute and ask your questions. Before that, yes, Bipin, thank you so much for joining in again, by the way, and taking out that precious time and taking out that important moment that, yeah, you need to address concerns which the nutritionists have been asking for. Thank you so much for that. So, Bipin, what do you have to say for everyone who has joined in?
Thank you for having us, Amarjeet, and it's been a pleasure. I think your enthusiasm and your quest for knowledge continues to, drive us in many ways. What Renuka said about her being slightly nervous stems from the fact that we all know that we don't know enough. And while we look at data, we understand a lot of things that data speaks. I mean, it's a gold mine. I mean, when you look at when we sequence the microbiome, when you sequence somebody's gut microbiome, there's a gold mine of data out there. Do we have all the answers? We don't. Together, with the folks that you've brought in fact, I would say the functional medicine fraternity is probably among the most well-equipped to work with things like this. So together, I'm sure we can do phenomenally well in helping people get better.
Very rightly said. Very rightly said. And with that, Renuka, I believe you have something to share with the participants. So we shall go ahead with that. You can. I believe you have the options to share the screen.
Yes, let me just try that. Thank you. Could you just enable sharing, please, Amarjeet?
Yeah, just a minute. Okay. Can you try?
Yeah, I'm just trying. Yes. Is my screen visible?
Yes, it is.
All right, so here we go. Again, thank you all for being here. And in fact, you are the best audience to understand what we are going to be talking about today. Mainly because you understand, as we do, that everything in nature is interconnected. And so is our existence and our relationship with the gut microbiome or the bacteria that live inside us. The balance of these microbes actually defines our existence, our life, our mood, and even how we interact with nature. As human beings, we have been very perseverant about finding patterns in life and finding meaning of existence, finding meaning in connections. And at IOM, our passion has been to find these patterns in the crosstalk between the microbiome and us. And the hope and the understanding is that when these patterns are put together, we are actually moving towards better health, and even a better planet. So for those of us who were not here last time to hear what the introduction that Bipin had given. So what do we do at IOM? We are essentially a microbiome-focused company. We work on the convergence of the microbiome science, the details, the intricacies of nutrition. We leverage mathematical models AI, and we also borrow wisdom from the food part of Ayurveda. We have 3 products for our users: Gut Sense, IBS Heal, and Gut 360. I'll quickly take you through these products eventually. I'm hoping that you will have questions for me at the end of this presentation. Like I said, our prime priority and our prime interest is in not just making you healthier and prepared for life, but seeing if this can be a healthier world even for tomorrow. As I mentioned, we have 3 products currently that we offer to our users. Gut Sense is to enable good sleep, increase energy levels, and of course, reduce stress. IBS Heal is for the symptoms of IBS, constipation, diarrhea, plain bloating. And again, like I'm preaching to the choir here, all the symptoms of the health challenge. And Gut 360 is more of an executive health checkup equivalent where we look at nearly 28 to 30 conditions. So one of the prime things about a health journey, and you are the best audience to understand this, is the entire value chain that a client or a user might be coming to you with. So what we offer at IOM is the entire journey from awareness to preventive healthcare to a situation that is just short of the disease, as well as the care continuum. So most of our products are able to transfer their value across this entire value chain. Bipin, please do jump in if you feel that there is something that is needed to be added.
Please go ahead.
Allow me to introduce the first product, Gut Sense, which is focused on ensuring better sleep, better energy, and lower your stress. So what does the user actually get in this journey is, one, the bacterial composition and its impact on what we call sense or sleep, energy, and stress. Now, this is where our core patented technology comes in, which is the optimization of the bacteria. Basically, what our optimization technique does is not only does it define the bacteria and form a network with the conditions that that bacteria is impacting in you, it also gives a direction to the bacteria to be moved up and down to ensure that your health is at the optimum. We also do a current health summary of your gut to define, is there any other imbalance that is actually impacting the gut? And as functional medicine practitioners, you would understand that the impact of health is not single factorial. There is generally a multitude of reasons that impact the gut. So just sleep, energy, and stress can have a multitude of factors, even within the gut or the gut balance, that can be actually influencing your health situation. Based on this information, we create customized microbiome-based food charts, which has also got a complete buy-in from the nutritionists that we have on our panel. We include doctor consults, we have meal plans. Obviously, this depending on how we work with you all, this is tweaked to make sure that the best impact is given to our users. Now, these meal plans primarily are based on the bacterial optimization. They're based on the microbial composition. They're also based on what is the required nutritional recommendations for the person at this point of time. This takes into consideration their tastes and preferences and what their current health condition is. What's very important for us, and as it would be for you, doctors, would be a feedback loop, which basically not just helps us understand how the user has moved through the journey, but also how we can make the system also understand and get smarter as it goes on to provide further services. Typically, this takes the follow-up is for about 6 weeks. The report takes about 4 weeks to be delivered to the user. I'm just giving you a quick peek through into what the report looks like. These are little snippets from the report. Happy to take questions on the report, the service at the end of this presentation. As I mentioned, on the extreme left, you look at these little thumbs up and thumbs down. Basically, these are a summary of the gut to say whether there are other factors. Maybe there could be an excessive amount of protein metabolizers, an excessive amount of bacteria that are producing lactic acid. or there could be a shortfall of lactic acid. So these are prime indicators that we look for in case that is impacting the sleep energy and stress. We also do a direction indication of which are the bacteria that we are addressing, which are the ones we are moving up, which are the ones we are moving down. Now, one of our first concerns has been typically people associate bacteria with illness or a health condition. You think they are disease causing, all bacteria are disease causing. And interestingly, it is not so. These bacteria are extremely vital for our overall functioning, and they perform multiple roles. They could be involved in several metabolic pathways. They could be catalysts, or they could be producers of several SCFAs that we require. Having said that, a large part of our work so far has been to educate the user that these bacteria are not disease causing. They're actually helping your system. However, at that point of time, a remodulation of these bacteria would be optimum for you. And that's what our the takeaways from this report come to, which basically translates into what one should be eating and what one should reduce eating. As functional medicine practitioners, you would understand that gut diversity still remains one of the prime metrics of what a good gut should be like. One would say that when you know that the more variety of food you have in your gut, why would you stop a person from consuming certain foods? The objective here would be to not stop any foods, and that is why the microbiome approach becomes a part of a more inclusive diet rather than a restrictive diet. We are saying include all the foods in your diet, but don't and increase a certain types of foods, reduce a certain variety of foods, just to make sure that you're actually encouraging the right bacteria and discouraging the bacteria that are not helping you at this point of time, yet maintaining gut diversity. Moving on to our second service. This is IBS Heal, primarily designed in association with doctors, gastroenterologists. We understand that IBS is one of the prime pain points of doctors as well. It's a confounding problem, and we have to admit that it has not been less confounding from the microbiome side as well. There are multiple signals that come from the bacteria, and even something as simple as constipation doesn't have a single definition. To differentiate between constipation, functional constipation, or constipation caused due to any other reason has been very tricky, and that remains so. However, based on the multiple studies, hundreds of studies that have been done internationally, we have tried to narrow it down and bring some value to the doctors to say that in case this IBS is because or does have a correlation with the microbiome, maybe this is a challenge that can be addressed also through the microbiome. So what we assess here is what is the bacteria in the gut and what is the influence or what is the signal that they are getting from the microbiome to that particular symptom of IBS. So we do a full gut health summary. Now, this is something that we do, which is very interesting, which basically is called the metabolic flux modeling. And I'll just come to that. Maybe Bipin can help me explain this a little better. But primarily, we please go on, Bipin.
No, please go on. Please go on. I was just...
Right. So the system that has been developed and what we use is to see if the metabolites that these bacteria produce, metabolites, again, preaching to the choir, is chemicals that are produced in the body, and these chemicals can be produced by the bacteria. And if these bacterial colonies, when they are increased or decreased, are they actually having an impact on the metabolites that have been produced? And that is the ultimate aim of increasing or reducing these bacterial colonies. Of course, IBS is a condition that we believe is best treated by doctors or functional medicine practitioners such as you. And this process has been made primarily to be an additional tool for doctors to be able to give extra information and probably narrow down the therapy that they offer to their patients. The objective also would be to improve the quality of life. Typically, and we know that most doctors recommend either probiotics or they recommend a low FODMAP diet, which eventually starts affecting the quality of life of most of the IBS patients. Our intention would be to see if that quality of life can be made a little better. And we are not yet there where we say that we can solve IBS. Our first level goal would be to see if a person has pain 8 times in 10 days, can we bring it down even to 6 times in 10 days, and then take it further from there? And if they are losing 1 day a week, can we make it losing 1 day a week, 1 day in 2 weeks? So that's where our goals lie. So just to show you what the report looks like. So again, we do initial indicators. A lower gut diversity is often associated with a disturbed or a dysbiotic gut. Similarly, a bad or a low firmicutes to bacteroidetes ratio is associated with a bad gut. We have a basic level of information that we do share, which is on what are the bacterial pathogens. We understand that IBS is in allopathy or in modern medicine as we know it, not directly connected to bacterial pathogens, and it could be an offshoot of a previous gastrointestinal disease. However, we have had a requirement from several doctors that we work with that we would like to check if there are bacterial pathogens, even something like a C. diff or a Helicobacter, which might be lurking at the backside to see if that is having an impact. Similarly, we also check if there has been an imbalance because of low probiotic bacteria in the gut. Most of these are based on the algorithms that our team has developed, which talks about what is the relationship between the microbiome and IBS, and also detailed insight into the bacterial connection with every single symptom. Now, in this, you could see that, I mean, this is a sample report, but you can see that the bacterial impact on bloating and flatulence is very strong. And this person has a fairly moderate connection with IBS diarrhea and the microbiome. This basically gives a direction to the practitioner to see if by addressing the imbalance in the bacteria, can we make this person healthier or at least move towards what we believe is a less imbalanced gut. Coming to Gut 360, like I said, it's more like an executive health checkup. We've had several users of ours coming back to us asking for a wider view of their gut. What we have done so far is 2 services which go deep into the gut for a particular condition or a particular health area. Here we look at 6 health areas, almost covering about 28 to 30 conditions, which includes digestive health, metabolism, respiratory health, heart health, emotional health, and nutrient synthesis. So this gives a very wide connection. One of my favorite examples is a young lady who had come to us and she says, I'm not able to lose weight. Despite all the exercise and despite all the weight loss programs that I do. And that's also because there has been an impact of the bacteria. The hope being that you address the bacteria and maybe your body becomes more amenable to the external changes that you're doing. Typically, all these 3 services take about 4 weeks for the report to be generated, and then we hand over the user or the patient to the doctors. So this is what the reports overall look like. We again look at the gut diversity, and as I had mentioned, it's one of the prime metrics of a good or a healthy gut. And we go through every single health area to see how the person is responding or how the microbiome is responding to their gut. Again, one of the very interesting persons we had a high number of bacteria connected with, reflecting on asthma, and she would tell us, I don't have asthma. However, what we realized is that she is a very heavy smoker. Now, the body responds like that it is being invaded by something that it's not very happy about. So the body might be responding to the discomfort it feels and responding almost similarly that it would in a situation like asthma. So Bipin had mentioned earlier that do we have all the answers? We don't. What we are trying to look for is to find a deeper understanding into the connections between the microbiome, hoping that we can correct the imbalance. So what we are working on is, and I'll explain, I understand that there were a few questions on what are the different sequencing technologies that we use. And this is something that has been very interesting for us because this is what we are working on. Right now, we are using 16S sequencing, and we are also working on our pipelines for whole genome or shotgun sequencing are ready. But this is the extra information that you get, one on, of course, the top abundant species, the pathogens at a species level, and of course, virus, fungal, all that, and even the probiotic bacteria. Now, why this is useful is that one could just pick up, say, in this case, if you look at the probiotic bacteria mentioned on the right side, you could just pick up a Bifidobacterium longum and say that this person has very, very low Bifidobacterium longum, and maybe
Renuka, just sorry to intervene. The slides, I mean, we are unable to read the slides. That's what the input has come in. So, would it be okay?
Sure.
Yeah.
Is this any better?
Yeah, I believe that's fine. Okay, and Bipin is saying that the slides and the reports will be sent. Perfect. I believe Dr. GK, I think this is what you were trying to ask, right?
So
Great.
this is more of an indication. Happy to share the
Thanks.
reports with you. Happy to share this deck with you as well, Amarjit, and all of you here at YellowSquash. This is more of an
Thank you.
indication to say that this is designed in such a way that a doctor can actually pick up these bacteria, and these are commonly available probiotics. These are approved by FSSAI. You can probably just pick up a Bifidobacterium longum or a Bifidobacterium bifidum and actually administer it to your client if you feel that that is what is required, because you know, based on this report, that it is low for him or her. Similarly, if you do see a virus, or you could see Erysipelatoclostridium, also known for gut inflammation, any of these bacteria, happy to share the information as well. What we are looking for from all of you here is also things that you would like to see in these reports. This is while we have already gone to the user, a lot of changes have happened because we do have the information, but what makes it helpful for us to see if we can keep working on it to make it more relevant for all of you. Do I see more hands raised, Amarjit?
Oh, we have a couple of questions in the chat box already. However, I believe you have given an overview of everything. Thank you so much. That seems to be quite a lot. And I'm pretty sure many of the questions might have already been addressed. And with that, are we open for the Q&A, Renuka?
I'm just addressing this last, I think, 1 or 2 questions that had come in. Amarjit, you had shared it with us on what is the difference between a qPCR, which I think a lot of you are already using, and what is the difference between what we do and what we hope to do.
I believe you're talking about the sequencing, the RT-PCR. Is that the question that you're addressing?
So they are connected technologies. qPCR is specifically being done, and I think it's being used by some of the doctors here. I think this is the GI mapping that we had been asked about. GI mapping uses a technology called qPCR. And the question that we had been asked, for those of you who are not aware, is which technology should we use and why? Now, here's what we believe. At IOM, we are fully equipped to use 16S and whole genome or shotgun sequencing. The benefits are placed in front of you. Now, none of these technologies have either a superior impact or an inferior impact. It is more of a advantage that you are looking for. For instance, a qPCR primarily looks at a quantification of known microbes. There is a set of microbes that you're looking for. There is a set of conditions that you're looking for. The person might have, say, IBD, or the person might already be coming with a previous Helicobacter infection. So the doctor is well aware of what he wants or she wants, and that's what he or she is looking for. In 16S and in whole genome, we basically look at the entire community of bacteria, which means that no bacteria is left out. And in whole genome, you also add in virus and other fungi as well. And it also gives you the metabolic pathways. So either is this procedure better than the other procedure? It depends completely on what the doctor is looking for to provide to their client. There was another question which I'm just addressing based on what does IOM really do. So what we do is we form these networks between the microbe, the disease, the metabolome, the food, and this is based on the AI that we have built. This particular network that we have built also takes in the metadata. So while we believe that there is a lot of feedback loop that is constantly going into making the system stronger, we have already built a whole level of this platform has been built in such a way that it can take on and actually give out extremely interesting insights. And just to give you, we can go for the Q&A right now, but the objective being that we are primarily looking at patterns that can bring more value to all of you here.
Thank you so much, Renuka. That was a wonderful session there. Bipin, do you want to add something before we start off with the Q&A?
Renuka's covered it quite well to give people enough knowledge to ask more questions. I think she ended it off also quite well in terms of that, it's not one is better versus the other. It depends on the context. It depends on the context. It depends on how you want to pursue it. But what we do know, because of the way the technologies are rapidly accelerating, is that when you get to know the entire set of microbes, fungi, etc., in your gut, based on technology today, you can start deciphering and making inferences that you would possibly not have otherwise. For example, I mean, typically when you do a qPCR, like Renuka said, is you check for known genes. You check for probably about 50 different microbes, fungi, etc. But having, I mean, for example, one of the people that we had was a person who had dysbiosis, because I think he had some condition earlier, and he was asked to take lactobacillus, and he kept taking lactobacillus to a point where the dysbiosis was caused because of excessive lactobacillus. You wouldn't get to know such things in a qPCR. But whereas in a qPCR, you would know that this person's got more H. pylori at a granular level, and therefore you could possibly take steps to address that. So it's a question of figuring out what works, what doesn't, what works for the person in the best possible manner, and at the same time also realizing that there are technologies evolving. Yeah, so we'll take questions.
Yes, certainly the exciting part for everyone. Yes, Haji, I can see you have raised your hand. Please go ahead.
Yeah, 1 second. So my question here was, I just wanted to know that your test charges also include deep dive consultation.
That is correct. However, we have created separate packages for people at your team at YellowSquash, because you would have a different requirement. So we are happy to break it into modules and give it to you as per your convenience. I think we have separate prices for YellowSquash.
That is one part. The other is also that we know we have to work with the medical community, with the nutritionists, with health coaches, with fitness experts. And there are some levels of personalization and precision that they would want to add to based on the way they approach health. So while we can give certain recommendations and we can be absolutely certain that we want certain pieces included for the health, it's up to the partner to decide finally how he or she would like to take it forward. So that level of adaptation, modification is possible.
What we offer essentially is a framework on which the end result or the outcome, you can choose to take the entire plan, or you can choose to take the framework and build further based on your therapeutic preferences.
That's great. Thank you for that. And I have another question, like regarding the peri and postmenopausal women. As you were saying that there are IBS and other related disease, microbiome testing is happening with you people. So do you have anything to do with the peri and postmenopausal women also? Do you check on that? Because normally we see that suddenly in post and perimenopausal women, there is a lot of weight gain and gut issues happening, and obviously the low estrogen levels are affecting the gut microbiome. So how do you like majority of the women come for that, and the conventional medicine, they say that it's just in your head, just eat less and work out, and that's what they say. So how can we help perimenopausal and postmenopausal women through your testing?
I see Bipin smiling, so I think I'll let him answer this one. Go on, Bipin.
No, please, it's up your alley. Please go ahead.
Ma'am, so peri and postmenopausal, there has been a lot of research and study that has been done on the connection between the gut microbiome and specifically, as you said, on the estrogen levels. We are still working on this facet. It's of deep interest to us. We hope to have it as probably in the next quarter or in the quarter after that. We hope to have a service on that as well as soon as our science is completely there.
Okay, so you're working towards that, hopefully. That's great.
Yeah. So in fact, what you have highlighted is also an area of differentiation in some of the technologies with NGS, which is where 16S and whole genome come in. You get to see the entire spread of microbes, right? And now we know the bacteria and other beings affect so many health conditions, including the production of different pathways that affect perimenopausal women, postmenopausal women, and even in the women's health, maternity, post-maternity, all these other areas. That it's an exciting field to make some of these connections, insights, and finally offer these in therapeutic manner, which typically you would not get in a qPCR kind of a methodology.
Mm-hmm. So do you think then comprehensive stool testing will be the thing for that that condition?
I still go back to the context. It depends on what the context is that you would like to apply in terms of the lens that you want to put in.
Okay.
At the moment, I would say both are useful, important. Eventually, I think the way NGS is the velocity of change in NGS, the velocity of improvements in NGS, I think will eclipse the other pieces.
Okay. And I have 2 more questions to ask. Do you provide any training for the practitioners to understand your tests?
Yes,
from scratch. From scratch.
Honestly, ma'am, we have had most of our practitioners already fairly up to date with the science, really. But happy to go through with some sessions, explaining our report, walking them through the report, and even having personalized sessions with the doctor before the doctor delivers or administers the report to their clients. Happy to do that.
In fact, to add to what you said, Anuka, I think we should work with YellowSquash to to evolve a program to do something like this.
Yeah, I would love that. Yeah,
sure,
looking forward to that.
Definitely reiterating the things, all the professionals who have joined in, we are definitely looking forward to equip you with the entire arsenal that you need to actually attack the world of chronic ailments. So don't worry about that. If something of that possibility is something that you need, we can definitely work on those lines. That's not a problem at all.
If there is anybody else asking question, I will hold on my question, or shall I ask?
Yeah, I think you had the question which was posted in the chat. Am I right? The stool
Yes,
sample.
I'm asking the same. Yes.
Yeah. So
the
as a
other
stool
the other one is.
sample, I believe you have read that question, Bipin. As a stool sample, it'll have 100 times more bacteria than the intestinal microbiome. How do you establish the diversity and the number of gut flora?
And actually, stool sample is representative of what happens in your gut. I mean, that's how we take it. But there are specific sections which offer denser levels of bacteria. There are specific areas, and those are all computational extrapolations that we make. The best, the possible means would be going and picking up a sample and saying analyze the sample, but in most cases that's not really possible, is it? So and that's why computational techniques are improving. That's why computational techniques can be steadily and make the difference. It is in saying, if it goes through a part where the mucus layer, the lining, doesn't have the facility to pass on certain bacteria, then and you know that certain bacterial colonies reside in these areas, then you make that extrapolation to say that in this bacterial region, in this region, these bacteria live, and possibly don't expect more than that. If you take one, it's a representative of 100. Whereas in some other areas, some other bacterial colonies, even if you take 100, maybe it's a representative of 100. So these are extrapolations that we make and work from in best cases, in a best case scenario.
Can you shed some light on your toolkit?
Of course, I mean, maybe we can show you.
Yeah, please. What all will be there? How the person has to? Will it be? They can send you by post and all that thing, please.
Yes, just to quickly run you through the process. I'm afraid I hadn't included that in the.
I'll go grab a kit.
In the meantime, you can do that. We'll take another question in the meantime. I believe everyone would be interested in this. So by the time Bipin arranges it, Renuka, let's address the next question which Anjali had asked. After that, we'll take up Suchna and then Venkatesh's question. So Anjali had asked in the chat, how is it possible to list out all disease-causing microbes with the disease they cause, along with a list of probiotics, along with suggested dose to correct the same? In 3 columns, all made to the participants. Would love to study your sample reports and connect this info and accordingly send our patients to you for testing.
That's a good one. That's from
Anjali. Anjali, yeah.
So Dr. Anjali, what we do currently is we follow the standards of definitions of what are pathogens. These pathogens might cause a multitude of diseases. For example, I mean, and there are certain pathogens that cause only a certain kind of disease. Like an E. coli can cause a I mean, it can go from gastritis to diarrhea to a whole range of gastrointestinal inflammations, resulting in a whole lot of infections. Whereas there would be other bacteria that could probably cause just one infection, or they could be bacteria that are associated with several hospital-induced infections. So what we do highlight in our report is a list of pathogens. We also highlight what is the abundance in that particular user. We would not yet be in a position to say, because this is different from a stool test that is done at a hospital or the culture test that is done at a hospital. In the culture test that is done at a hospital, they look at the rate of growth of the bacteria in a particular sample in a medium. Now, we don't do that. We are only giving you the current abundance of that particular bacteria. Whether that translates into a symptom in the user, you might need to do the simple stool test that is done in a hospital. Now, that's our intention is to find the entire bacterial community. So can we list the bacteria? Can we list the disease-causing microbes? Yes, we can. Can we list out the disease that they cause? Possibly not, because like I said, each bacteria causes a multitude of diseases. But these bacteria, like an Acinetobacter baumannii, it is known to cause several diseases. So we can list out the bacteria. We can tell you that it is certainly defined as a pathogen either by WHO or by UN. And can we give a suggested dose? I'm afraid that would come into your purview because that is more on the medicine side. We can tell you that this bacteria exists and we can hope that you will take it forward.
I believe there has been a little technical
But
glitch at
now
run.
this is the recommended course of action.
Yeah.
Okay.
Am I audible?
Renuka, I mean,
Yes.
Am I audible?
Yes,
Yeah,
you are
yeah.
now audible. I believe, Anjali, you want to speak something. Sorry,
Yeah, I
Vipin, just a minute.
yeah, just I was just responding to Renuka. Thank you for your answer. What I actually meant is that, you know, we would, so this kind of listing would expedite the usage of your gut microbe tests. Because otherwise, you know, of course, one knows that there is a disease connect and which bacteria cause what, and roughly you know the probiotics. But since you're in the business and you know exactly, you've done a lot of research as to how much dose is important for what kind of bacteria. I'm sure you have research papers on that, right? So if you could share of that, because otherwise to learn the whole thing, of course, we are open to learning, and I'm glad you have a whole, you know, team which is ready to teach. However, this kind of sharing, what I've asked for, will just expedite the whole process. You know, once you know, okay, this is the bacteria, this is the disease, these are the rough you know, of course, we are not going to follow the dose exactly like how you recommended, but it gives you an idea, you know, just to get an idea to expedite this whole thing. Otherwise, people take 6 months, 1 year, and by the time you've just forgotten about, you know, this, and you're doing it in your own mom and pop way. But a list from you would help all the participants to plunge into the testing. That's what I meant. So if that's possible, it's lovely, but thank you for your answer.
Yeah, may I see, the thing is, Anjali, we need to shift the thinking that it's only pathogens that cause.
No, obviously.
Yeah,
there is food, there is stress, there's toxin. Of course,
no,
we know all of
that.
that. Yeah.
Yeah, in the sense that it's only that pathogens cause certain disease, or like you mentioned, disease causing. Those are disease-causing microbes. There are a set of pathogens. But diseases are not caused only by pathogens. Diseases or conditions are not caused only by pathogens. They are caused by some bacterial. So this is the shift that we all need to get into. So there is a bacterial ecosystem. And I think I mentioned in my last thing, and my team hates me for saying this, but it's like dogs and cats and everything else. If you increase the number of dogs, typically what happens is the cat bacteria kind of gets low. That's possible.
Agreed. Everyone's aware of the firmicutes and the bacteroidetes and the dysbiosis.
No, it's not just that. So what I'm saying, there's a pathway to certain production of certain metabolites. So if, for example, there are some bacteria that consume or ferment milk that you take, and typically, you know, that's what you look for. I mean, in the production of milk, you'd say, hey, is there lactobacillus, you know, or some variant of that processes milk. But what we didn't look at in microbiome version 1.0 was to say that, hey, there is lactobacillus to ferment the milk, but is there the second set of bacteria that consumes the output of what lactobacillus produces, which is possibly sulfur? And is there a bacteria to consume that sulfur? If there is no bacteria to consume that sulfur, then, you know, that's when you pretty much have bloating and flatulence. But if the value chain was perfect, you wouldn't have. So what I'm getting to is it is not saying that, not looking at bacteria as disease causing, or as ones that are responsible for certain things. It's possibly a community. It's an ecosystem that needs to work perfectly in sync. And that is also where, Renuka mentioned, maybe we can't even say dosages unless we look at the whole ecosystem.
Okay, let's forget about dosages. Okay, you know, the rest of it, I'm just saying, you know,
it's
so you make
what I'm saying.
Yeah,
it's too vast to find the combinations and give you a list saying, you know, this is what it is. Maybe for specific, see right now, and that's the thing, right? There are 31 bacterial species strains that are approved by the government of India to take as probiotics. Is that sufficient? I think the answer is a no. So if you were to quantify it, saying, you know, for these diseases, just take these bacterial strains, then it's a very simple answer. But it's a little more complex than that.
No, I understand, but you already have 3 types of reports in which you have already done some homework for 6 diseases or 10 diseases or 26 conditions, etc. So for those, you have your answers, right? These are the probiotics.
No, it changes, Anjali. That's what you need to look at. It changes. Each person doesn't get the same, which is where, see, I think you're also coming from the point of the qPCR piece, where you just look at 50 different bacteria. That's not how things have been evolving. That's not how things are moving. You need to look at larger pieces, which, you know, doesn't come in when you start looking at things only these 50s, possibly more.
Yeah, no, we were looking at a point to begin, right? We're not asking, for the entire world. Obviously, that's a large world of the, microbes and that's, but so there's a point where you begin and you begin with 50, you begin with 30, you just begin with whatever knowledge there is, and then you build on that. So that's faster than, doing the entire training and then waiting and then kind of finding it on your own. Of course, one can do it. But it was just a request, but that's fine. You've given me the answer. Thank you.
Thanks, Anjali. Just to add on a little thing, while we were having an internal discussion, I had shared a sample report.
Yeah, we've got
And
that.
over there, it was very much evident that, there is no good or bad bacteria. Suddenly a good becomes a bad because it's just too much,
Yeah.
or we intentionally need to reduce it. And so it becomes pretty customized. Look into the sample report. In the sample report, you'll have each of the bacteria and its characteristics, why we are thinking of reducing it to optimize the final microbiome, and why we want to increase this to do what. It comes in that. It comes one by one in the report. So using a customized report gives you a better answer than a generalized chart that might tell, okay, this bacteria does this, and this is what you should do about it. A report itself can give you a better
No, obviously.
picture of the thing. I hope that
Yeah.
answers a little bit of what I'm
Yeah,
what you have been
no,
asking.
yeah, it's I don't want to waste everyone's time on this. So we do this gene testing. So they provide us a manual, okay, for all the genes that we test and different conditions. So there are lots of genes, and they provide us a manual. So it's like a guide. Okay, so then it becomes easy because we have large teams. We need to train the other doctors. So you if you have a manual, it helps you to begin somewhere, right?
Let's start with
Sure.
going with what you're saying and give it to you for the areas that we are really good at, which is energy, stress, sleep, possibly IBS, different conditions of IBS. Let's start with bacteria that can be connected to them.
Yeah, that's what I meant. That's exactly what I meant.
Yeah, perfect.
Top level.
Good. Yeah.
Let's see how we can do that.
All right.
Thanks a lot, Anjali, for that question. Pretty much a very important one as well. Thanks, Bipin, for answering that.
Thank you.
Thank you for holding on, Suchna and Venkatesh. So, Suchna, please tell me your question.
Yeah, hi. And
hello.
Thanks for the session. It was really great. Amarjeet, thanks for bringing this to us. My question today here is that like suppose somebody is suffering from a leaky gut, high levels of zonulin and all that. So, and C4, SIBO and all. Is there a way to assess whether a person has it from your tests? Any of your tests?
So, Dr. Patnaik, what we do is, I mean, we haven't included in the short answer is we haven't included zonulin. There are several studies that include leaky gut and zonulin to the microbiome. In terms of gut inflammation, as gut inflammation biomarkers, we haven't included that primarily because we went with IBS first, and zonulin, if I'm not wrong, and please correct me if I am, zonulin and fecal calprotectin are more on the gut inflammation or the IBD, IBC,
right,
on that direction. We do hope to include these in the coming services. So yes, so right now we don't have it. We can, and we will, I mean, at any point of time, most of our work is going on to include as many illnesses, but we decided to go to market first with these 3 services that we have. But that's a very good question, and it also gives us a lot of encouragement that there is a query on that.
No, right, because you know, even autoimmune cases and most of the cases who encounter inflammation have a leaky gut also. So if we could add something like a test where we can also find out whether somebody has a leaky gut and zonulin levels are high, it would be helpful. And for SIBO C4, I think SIBO C4 is it possible?
So
to find out.
So what we do, and again, we ride on the shoulders of the physician as well. When we share the composition, especially when we do a whole genome, we are able to see if there is a fungal overgrowth, or if in cases of SIBO, there are certain bacteria that are seen overabundant. The The stool sample that we collect is primarily a representative largely of the large intestine. And
Okay.
there are certain bacteria that reside in the small intestine, which we can figure out. However, we would recommend that there are other tests that can give you a better information. Now, one thing that we speak with our practitioners regularly is that we don't use the microbiome as a diagnostic. We rely on your expertise to diagnose a particular illness, specifically in case of IBS, because that is something that it's not at a biomarker level. What we do is we look for signals of connection. If a person comes to us saying that I have been diagnosed with IBS, then we look to see if there is a connection between IBS and this person's symptoms, and can it therefore be cured through repairing the imbalance. Would we say that every person with a low diversity and a high F by B ratio has IBS? It can be indicative of several other conditions as well. We use the IBS severity score. We use all these scores to kind of narrow it down to say, yes, the doctor has confirmed this is IBS. The person is showing symptoms of IBS, and the microbiome is also reflecting that there is a very strong connection, very strong probability that this person has IBS. Therefore, let's treat it through the food that they're going to be eating.
Yeah, okay. Thanks for that. And one more thing is your kind of testing all the three products, which one would you suggest for an autoimmune patient? Suppose somebody with autoimmunity comes to us and we want to find out more details. So which one would you recommend out of the three products?
Dr. Patnaik, what is the outcome that you are anticipating from?
If suppose somebody has Hashimoto's, we all know that like EBV is causing, or maybe and in autoimmunity, your gut is always imbalanced. So,
Yes.
which of the three, suppose you have the gut sense, gut 360, IBS heal. Is
Yes.
there any one of these products? Which one out of these would be helpful for an autoimmune patient? That's what I wanted to know. Like, suppose you want to recommend a test, which one do you think would be better for somebody with autoimmunity?
In order to understand the gut and its composition and how it's reacting with different aspects of your health, and like we said, in Gut 360, we cover almost 30 aspects of health. So that we would believe would be the closest to seeing whether there is an imbalance or whether there is a connection to this person's health. Do we particularly look at diagnosing Hashimoto's? Not yet. We do have a section that looks at signals towards IBD and IBS. And happy to include more in the Gut 360 based on a good amount of input from all of you. Currently, we would say that would be the closest to give you an idea about what is actually happening in that person's gut.
Okay, right. Thank you so much. Thank you. Amarjit, I'm done.
Thank you. Thank you, Suchna.
Thank you. Thank you.
Yes, Dr. Venkatesh.
Thank you for the good presentation, Renuka. Actually, it was very good because as I was telling Amarjit, I was unable to hear clearly the earlier talk.
Nice.
So actually, we were going through a sample report, and then we found that you're giving certain suggestions regarding the food. Okay,
Yes.
these food you have to take more, or this you have to take less, and all that. How are you deciding this, basically? Is it going to be a set of nutritionists who are sitting there and then doing it, or how is it done? Because any biases that get into it will always create problem in the final stage. So I'm more worried on that aspect.
No, you're absolutely right, Dr. Venkatesh. And that's a very good question to ask, because it basically talks about what is the outcome of all this testing that we are doing. Now, and if I may start at the basis, there are several factors that influence the gut microbiome. And the only factor that is truly in our hands at this point of time is the food that we eat. Could be stress in the environment, there could be pollution in the environment, there could be the kind of medication that you've been taking as you grew up, or you what kind of was it a natural birth? Was it a cesarean birth? All those things do impact your microbiome. However, the one thing that's in the hand is the food that we eat. There are certainly, and what our science has told us, and the kind of data that we have assimilated, show a very robust connection between certain foods and the preferences of the bacteria to thrive on those foods. So there are two things that determine the food recommendations that we give towards the end, especially in the Gut Sense report. One is, of course, the network that we have created between bacteria condition and the food that these particular bacteria thrive on. So that comes through an algorithm. You were asking about whether there is a nutritionist intervention in that as well. And yes, we do have a nutritionist intervention, and allow me to explain why that factor had been added in. Now, when we started sending out reports almost a year and a half back, we had exactly the thought that you had, Dr. Venkatesh, that the microbiome food list should be a pure, unadulterated microbiome food list. However, when we actually took it to our customers, that did not turn out to be very usable insights, because there are times when the microbiome, depending on the bacteria in the gut, would say that, it is good for this person to have, say, honey. There is a very strong correlation between honey and that particular bacteria that's residing in his gut. And we say, okay, have honey. However, this person is diabetic. And whether honey is recommended for a diabetic person or not, in the user's mind, it is that I can't have honey because I'm diabetic. And that creates a conflict, making it not a very usable report. So when we administer a questionnaire to the user at the time of purchase, the questionnaire basically takes into consideration their current health status as well. Are they diabetic? Do they have a thyroid issue? Do they have high cholesterol or any of these other issues? Are we going to the level of giving them medical advice? No. But at the food list, so the outcome is broken up into two parts. One is the food list, which forms the framework, saying that here are the bacteria which need to be increased. Here are the bacteria which need to be decreased. What are the foods that this bacteria will thrive on? What are the foods that will discourage this bacteria? So that is the prime premise on which the bacterial food connection is taken off. And the nutritionist at that point of time only considers whether this person has a condition and that food is contraindicated for that condition. A more detailed, a more thorough intervention of the nutritionist comes in only when the user moves to the meal plan stage. And that stage will depend on how we take this journey with YellowSquash. Has the customer opted for a nutritionist? Then we will offer that. If YellowSquash already has a nutritionist on board, then we will work with the nutritionist to take this forward.
Okay. So basically, my question is the food list, what you're creating for each of the bacteria.
Yes.
How are you deciding it? Is it based on the various studies that have been done, or how is it? How that list has been created?
Yes. So we have collated hundreds of papers wherein very robust studies have been done. One of the questions that we've been asked often, Dr. Venkatesh, is that this food list doesn't look exhaustive. It doesn't look like it has everything. And that's something that is difficult for us to create, because in case of a very, very specific local foods that is probably just found in maybe Bangalore, not just in India, there might not be very accurate studies that have been done, or there might not be adequate studies that have been done, which does not allow us to include it in our food database. So only when it adheres or it passes some of the stringent norms that our scientific group has made, once it has passed that, then it is eligible to feature in the food list. What could also happen is that one person might get, say, curcumin might be recommended for a person, and it just disappears from another person's list because our system and our algorithm did not find a very robust connection between that person's bacteria and curcumin. So the list that we give is not completely exhaustive, basically because it is based on global research.
Okay, I agree. Okay, then one more question I have. Basically, today itself, I found out there's one more microbiome company sent me an email
Yes.
They are also doing testing, but they are taking blood serum, and their testing costs are much lower. So I don't
Yes.
know. I'm not. This is the first time I've come across this. So I thought, can I have your viewpoint on this?
Yes, honestly, we are also slightly puzzled because um, there isn't a lot of research that is available for the blood microbiome. And to determine the blood, to determine the gut microbiome through a blood, uh, we haven't come across many research papers. What is promising and is again a newer way of analyzing the gut microbiome through the oral. Even that is a very nascent field. There is a lot of, to analyze the gut microbiome through the oral is a new field. The oral microbiome itself is a different form of checking for health. But blood, we still do not have faith in that science because there isn't enough. And if I were to ask my science team, they would say there is no paper out there that says that there is a way of finding the gut microbiome through the blood. But if this team has found it, then there certainly could be a lot more done in that area.
Honestly speaking, doesn't make sense when you are talking about the microbiome and the blood. Oral,
No, Haji.
fine.
Haji, I will send you the details to you.
Yeah,
You can
I
go
can
to it.
explain that. You know,
So there's a company, Microbiotech. They have
yes,
started this. They are they are actually working with some research people. Okay, some 2-3 research institutions in India, and already started doing the testing. So you can send your blood and you can get your microbiome tested. They will also
Maybe
give a full
they're
report.
correlating. Maybe they're correlating it than the microbiome actually.
They have some paper.
not maybe, they're definitely correlating. See how stool
Yeah.
sample works is there is, you
No,
find
like Renuka
microbial
said,
genes.
maybe in a nascent stage, but let us wait and see how it goes. So just wanted to just get a viewpoint from them because they are there
And
in the field. We are not in the field.
yeah.
It's
And maybe
extrapolation to a different degree. See what
just
happens.
a thought. Just
Please
a
go.
thought. Maybe it will help with the leaky gut issue, where there is a lot of.
How do you propose that? What's your theory behind that?
Obviously, leaky gut, lot of remnants and the metabolites do sneak into the blood, right?
Yeah, and that's the rationale, I guess. That's the rationale. That in the blood, then you would have to have some metabolites that are present in the blood, which was representative of what certain bacteria could be producing or not producing.
So it is better to go for a stool test than the blood test, I suppose. I don't know. As Vintesh is saying, let us see how it will go.
Yeah, the stool and oral and skin actually looks at the DNA of the bacteria that is there exactly as is. And some other extrapolations can be made once you detect that. But like Renuka said, it's possibly too early to say, can we detect the presence of all metabolites that are produced by different bacteria in the blood? And I don't think
really
there
come
is.
across that. We haven't
I
really come
really don't know. Just wanted your
Yes,
viewpoint.
But it's a lot of good science happening. So let's hope something like this
A
can
lot of
evolve better.
research going on microbiome.
Yes,
Yeah.
it's the new
Correct.
buzzword.
I believe even Dr. GK has said the same thing, as we always say, let's not comment about a science that we are not fully aware of. Learn both of them and then decide is an important thing. Definitely respecting all the things coming in. Very nice. I can see some very good engaging questions coming in. Anybody else has a question? We are ready to take the questions. And in the meantime, I'll just see if there is anything
We're waiting
else.
for the test kit.
The test kit has to be shown. Yes.
Yes.
Let's go ahead and just a minute. I would also like to throw a spotlight on you. Great. Okay.
So this is what the kit looks like. I mean, we've been economical and practical in that sense. It's not a really big piece. It's the size of your phone box. You unbox it and you have a set of accessories that you use to collect the sample. You have a self-addressed envelope that you can seal it and send it. And the most important piece is the vial. which can store the biological sample for up to 6 months in regular temperature. This is your DNA stabilizing fluid. That makes up the contents of the kit, really. Simple, not very massive, easy to use, and to the point. That's it. This is what the kit looks like.
Okay.
So
It
the
will be couriered to you.
instructions,
Yes,
This is my hand, so you can see, and I'm not very tall, so you can see how big it is.
So it comes with an instruction manual as well. It has a little collection carton for you to collect the stool. It has gloves and wipes and all the hygiene products that you would require to clean up, a little garbage disposal bag as well. And it has a little scoop and instructions on how much to put into the bottle and shake and pack it. It's delivered, this kit is delivered when a user buys the service. The kit is delivered to their house and picked up from their house or any area of their preference.
Got a home light box, so you can see what I'm not sure this is something that you all are familiar with. So you can see sample representation of the size. That's what it is.
Thank you. Thank you so much.
all right, what's the minimum sample that is needed?
So that's a good question. We generally ask for 3 scoops. It does get a little tricky when a person has IBSD, but what we have realized is that this vial that we are using, it's a 30 ml vial with about 8 to 10 ml of fluid. And so that about 3 scoops of stool sample and shaken well is good enough for us to detect what we need.
So what about if he has diarrhea or something like that?
Yes, in that time, we do request them to add in a little more. It does get diluted. In extreme diarrhea, if there is an infectious diarrhea, then we ask them to avoid, because these are biological samples and it might actually, in case of any accident, it might actually spread disease. So in case of an infection, we would avoid doing a sampling in the first place. Otherwise, we ask them to put in a little bit more.
Okay.
Question, Dr. Ganpati, also was, how do you handle if it is overdosed? Could you please explain that to me?
Dr. GK, why don't you unmute and ask your question? I believe his question is, how do you handle if it is if the sampling is overdosed?
You mean the sample? Is that what it is?
Excess, if that excess sample is added, how will you handle it? And do you do a pre-analysis before loading in NGS? I'm not sure of that.
So yes, there is, honestly, we haven't had a situation where it has been overloaded. I think most people are not very enthusiastic even scooping 2 or 3 scoops. Asking them to scoop a little bit more is a little excess. We haven't had this problem, but it's a good problem to consider. We had it. So if you ask if there is a pre-analysis done, in fact, there are, I think, 3 levels of QC for a sample when it is taken to the sequencing machine. So if you're asking for, so as soon as the sample reaches the sequencing lab, they do a QC on it. So yes, there is an analysis, a pre-analysis done. And there is then 2 levels of QC that is done after the sequencing has been done. Does that answer, Doctor, the question?
All right, thanks. Perfect. So any other questions? Please ask your questions. This is your session. This has specially been organized for you so that your questions can be answered. It has been a very informative session, I believe, Renuka, and everyone's got a lot of information. In the meantime, yes, definitely, I have already shared the sample reports with all of you in our groups as well. And remember that you can always use the code YSHealth for an additional 10% off on the discounted prices of IOM. Okay, so use the code, share it with your clients, and the process is again the same. You need to go to the IOM website, select the product that has been recommended, and you need to log in, definitely, select the product and add that code YSHealth to get the additional 10% off. Okay, anybody else has any further questions? You can raise your hands. Okay, what database are you using to get the results?
give me, if you could give me one moment. Also, before we are right now using Silva and marker databases. Would that help?
But it's not Silva and Markup databases that it is in its original form. And this is where the work that the team has done has come in. It has been expanded to create our own set of databases.
So is it free or a paid library?
As in, we don't sell the database or the library. That's the question. It's part of our product portfolio.
No, I think what he's asking, Doctor, is whether the library itself is free. This is a commonly available library. This is a universal library. Yes.
Which is a universal library, in the sense Silva, that's a database, right?
Yeah. Silva.
Yeah. So the Silva ones are.
Yes, please go on.
No, that's.
All right. Anybody else with any questions? I couldn't see any further questions on the chat box. I believe we have answered all the questions. Just revisiting the same. All right.
Just a short message for the doctors here is a lot of our work is done and determined and made for you. So a lot of these questions that you're asking us has a lot of value in improving what we are doing. So even after this, if you believe that there is something that you would like to know, or like Dr. Anjali had said, having these lists that would make it very easy, or a manual that would make it very easy. These are really good inputs for us. So if you think of something, or you believe that there is something that your clients could benefit a lot from, please do drop us a line. I'm sure Amarjeet will help us coordinate with you all also, and probably fix a session with you all and see if we can create something specially for you and your clients.
Thank you. Thank you once again, Renuka, for that. Just addressing one more question. If you want to, how is it different from GI mapping and the comprehensive stool test? Because this was a very, very common question that was asked. Could you please?
So I did do that last sheet on GI mapping. GI mapping does use the qPCR. And to address that question, ideally, the qPCR has its own uses. It is used as a diagnostic in several conditions. The limitation, however, is that qPCR, or what GI mapping does, looks at a limited set of bacteria. It does not do a community profiling. Like I said, it has its extreme uses. Some could also say that it is more cost effective. However, if what you're looking for is a community level understanding of the bacteria, a qPCR might give you maybe 20 or 30 bacteria that you have selected, or 20 or 30 areas of interest, which is what is done during the PCR. Unlike what we do in either 16S or in whole genome, where you get the entire list of bacteria. And you also, like in a qPCR, you might just get the keystone bacteria or commensal bacteria and a list of certain pathogens that you're looking for. If there are other pathogens that are not there in your initial diagnosis or your initial hypothesis, you might not be able to catch them. This is very, very need-based. If you want to do a full community profiling, we would suggest go for a 16S or a whole genome. If you are looking for validating a certain hypothesis that you have in mind, and you are looking for specific conditions, you're looking for a Helicobacter pylori, please do a qPCR.
I think that is a very, very cleared question, very, very clear thing that everyone has got an answer on. So really nice. I think we have answered all the questions.
Can I add something? I just
yes,
read out.
please.
So comprehensive testing is to more to do with the like hormonal issues and gastrointestinal issues, all those things. I think it is overall, it tells us about the overall health. And when it comes to GI mapping, it's mostly specific to certain bacteria and all that stuff.
It could be certain bacteria. Yes, it could be certain bacteria. It could also be certain biomarkers that they have picked. I went through I did do a little bit of cheating before this call. I went through their list. I have gone through their website extensively as well, and they have listed out the conditions for which this would be ideally suitable. So if you are, as practitioners, if you are very clear that these are the conditions that I'm looking for, and these are the particular, like you said, doctor, you were looking for zonulin. So if you're looking specifically for zonulin, that's a great test to do.
What about the bug speaks test? There is one more test called bug speaks, right?
Yeah. Yes.
So can you, what's your take on that?
Well, they're very good too, and I'm proud to have them as competition. Proud to have them as people who are in this domain. At the same time, there are differences in what we do and what BugSpeak does. BugSpeak provides an NGS overall math equivalent. What we have done is go down to specific areas, which is where in the question that Anjali asked, I was quite clear that we have gone deep into the areas studying bacteria and bacterial metabolites that govern certain areas, sleep, energy, stress, IBS, for example. So we know if we have to address them, how is that we need to address them very specifically, as opposed to giving a top level view, which is also, to your question of perimenopausal, postmenopausal women. When we go down, we go deep into understanding the bacterial ecosystem and communities that affect that, and how we can address that by addressing the communities that impact that. So
that's wonderful.
It's the level of, so for us to create the solution for energy and stress took us about a year, because we went absolutely deep into it in terms of finding not just the microbes, but also not just finding the first order microbes, but the second order, third order, what we will call up to path length tricks. We have done something similar, or are doing something similar for each of the areas in IBS, bloating, flatulence, pain, IBS-D, IBS-C. So, and in some sense, in some cases, I think we've just got an approval to do clinical trials, particularly on a specific type of IBS-D. So, so it's the level of depth that we go in, I guess, makes us different.
Yeah.
We don't provide a top level. So you see in our products also, right? They're very specific.
Yeah, that's amazing. How old are you in India?
We just crossed 2 years, but the 2 years that we have has been built on about 2 decades of work that our Japanese counterparts have done. They've been working in this domain of microbiology and computational biology for almost 20 years now.
That's great. That's great. So where are you put up in India? Centers? Your centers are in Bangalore?
That's correct. We're in Bengaluru.
Okay.
Bengaluru,
I'm from Bangalore, so I'm highly interested.
Yeah, sorry, please go on, Bipin. But the service is not restricted to Bangalore. We service the whole
I understand.
of so,
I understand, because the package will be delivered and it will be collected. That's great. Thank you so much.
And in Bengaluru, we operate out of what is called the Center for Cellular and Molecular Platforms, out of the NCBS, which is the National Center for Biological Sciences. We operate out of the campus there.
Okay.
So you're welcome if you're in Bengaluru.
Thank you so much. Yeah. Thank you so much for the input. Thank you so much. It was great having you people.
Thanks, Haji. Yes, you want to say
thank
something?
You so much.
Yeah,
no,
yeah,
I'm just thanking you. Thanks for the platform. Thanks for clearing all our doubts and making us more enthusiastic in these microbiome testing. So we have the tools, we have the experts, everything is there, all the support is there. So it will be a great experience ahead. Thank you so much.
Time to make the nation healthy.
Yes, sir.
Let's make the microbiome healthy. Thank you so much indeed. Thank you so much for your cooperation, and again, thank you so much for your valuable time, your valuable inputs. It makes a lot of difference. It's a win-win situation. Definitely the experts can learn a lot more with your questions. Keep coming up with those questions. Keep your curiosity alive. It will make a lot of difference to all of us, and I'll be more than happy to connect you guys. So wonderfully organized and said said, thank you so much again for your valuable time. If there is anything else that we need to find out, do connect with me. Let me know what are your questions. Okay, I'm going to be there searching for your answers. Okay, and get you connected with the people who can answer your question even more wonderfully. Okay, so don't forget to do that. It was a pleasure hosting all of you. I believe I cannot see any further questions. There is a thank you. Sure, Dr. GK. Thank you, Radhika. Thank you once again.
Amarjit, sorry again. Tell me about YellowSquash, please. What it is exactly. I'm new to this.
So YellowSquash is a platform which actually is connecting experts with experts and experts with clients. So it's a marketplace. It's a marketplace for the integrated practitioner, the holistic practitioner who is trying to connect with clients and who is trying to connect with experts as well. So because health platform is an integrated platform, it's supposed to be, it's just not like a doctor-patient relationship. Doctor also needs a lot of support, coaches, nutritionists, fitness experts, physios, etc., etc. Everyone needs to be all connected. It has to be an integrated network, and the clients need multiple support systems. So especially when you're dealing with chronic ailments. So we provide sustainable solutions to chronic ailments with the help of experts like you. That's what YellowSquash doing there.
Thank you.
Don't go by the name. Don't go by the name. That yellow comes in, that yellow black comes in. As you can see, I'm wearing the YellowSquash rather. So yeah, the yellow black is our theme, and we are more than happy to connect people. As I said, create a common platform where everyone gets a chance to get the best information that they are supposed to get. So that's what we do.
Great. That's great. All the best.
Thank you very much. Thank you once again. Very, very good evening to everyone. Have a wonderful weekend. And I really look forward to hosting another expert talk with all of you. Do keep posting in questions to me. And IOM Bioworks, keep doing the fantastic job that you have been doing. We really look forward to getting even further information. I'm pretty sure, I hope this particular session was informative and gave you much more, those eureka moments. Oh, why not this? That you can actually take care of. Any last words for our practitioners that you would like to say? Well, yes, Bipin.
I'll say thank you very much. And we should act on some of these queries that were raised today, including what Anjali asked about some set of initial bacteria that they can know. But I think one of the reasons I think microbiome science has not progressed is these tongue twisting bacterial names. It'd be a good idea to get all of you to remember those tongue twisted bacterial names.
And you write the pronunciation also in it. This is how you pronounce it.
Yeah, so all that may be essential. And those are my takeaways. And yes, look forward to constantly engaging. Maybe we should put up a platform for coaching the fraternity at large as well.
Yes. In the on the long term, definitely this is something that we will be looking in for. Thank you once again, and I'll just check the chat. All right, thank you so much. Thank you, Suchna, and thank you, Anita. Thank you, everyone. Bye-bye. Have a wonderful weekend, and I'll see you in another session. Bye-bye.
Bye, Martin. Thank you.
Bye.
Thank you. Bye-bye.
Bye.